And I'm going to mute.
Good afternoon, everyone. I'd just like to welcome you all to today's edition of the HNRCA Monday Seminar Series. I'm the Associate Center Director at the HNRCA, Roger Fielding, and it's my pleasure to introduce today's seminar speaker, Dr. Nicolas Musi. Dr. Musi is a professor of medicine and the director of the Sam and Ann Barshop Institute for Longevity and Aging Studies, and also the director of the Center for Healthy Aging, the San Antonio Claude D. Pepper Older Americans Independence Center, and he also serves as the director for research programs at the San Antonio VA GREC program at the University of Texas San Antonio Health Center. The title of Dr. Musi's presentation will be Targeting the Gut Microbiome to Improve Metabolic Outcomes in Obese Subjects. I've known Nick for a few years now, and he trained early in his career in Boston, although our paths never exactly overlapped when he was in Boston, but I did have the pleasure of actually visiting him a couple of times now in San Antonio, once when he invited me down to give a lecture and also to attend the really nice meeting that the annual meeting that the Barshop Center puts on every year. And they put it on at what I would best describe to us Bostonians as at a sort of a dude ranch. So I got to ride horses, learn a lot about science, and it was really a great experience. So I'll always remember that, Nick. So thank you for that. So without further ado, I'll turn over the meeting to Dr. Musi and would look forward to your seminar. Thank you.
Thank you, Roger. It's an honor to be here to giving this presentation, and we'll have our meeting someday again in Bandera, Texas, in the Dude Ranch. Can you all see the presentation?
Not just yet.
Okay, let me share it then. Share screen.
All set now.
Okay. Thank you. So my research is related to the effect of aging on metabolism, metabolic pathways, and also the pathophysiology of diseases, metabolic diseases seen as we age, shown here: obesity, type 2 diabetes, diseases of glucose metabolism, and also diseases of protein metabolism, such as sarcopenia. And of course, these are all problems that our older population faces, and they're major causes of disability and morbidity. Also interested in studying the interaction between these metabolic diseases. For example, this is data from a longitudinal study called SALSA, in which Dr. Jesuda Espinosa enrolled several thousands of older adults, healthy older adults here in San Antonio, and they followed them to see what predicts the development of frailty. And it turns out that in this cohort, sex, so being male, it was major determinant, but the second one is the development of diabetes. So glucose metabolic dysregulation preceded the development of frailty. So there is a connection there, and we're interested in studying it. And one of these potential factors that connects these metabolic diseases is, as you know, inflammation, which is thought to be underlying aging, but also many diseases of aging. So we've been studying for many years the basis for the inflammatory state on metabolic diseases and in aging. And we developed this a model that we've utilized to test different hypotheses related to inflammation and metabolic disease. To the right are different factors that would increase inflammation. So this would be, for example, a cell. It's a cartoon of a cell, could be muscle cell, although also potentially hepatocyte. And in the middle is the NF-kappa B pathway, a key modulator of aging that increases, a key modulator of inflammation, and of course increases with aging and also metabolic diseases. So we've been studying what are factors that drive NF-kappa B and other inflammatory signaling pathways. Of course, one of them is cytokines such as TNF. We spend a lot of time studying how a lipid accumulation inside the cell driven by high fatty acids stimulates these inflammatory pathways. Similarly, mitochondrial dysfunction and increased ROS. We've been focusing quite a lot on the potential role of toll-like receptor 4, shown here, that can be stimulated by fatty acids, but also, as I'm going to spend a fair amount of time talking, can be stimulated by endotoxins in our system. And then to the left are consequences of increased NF-kappa B signaling and increased inflammation in the cell. And one is inhibition of insulin signaling, leading to insulin resistance. Also, working through AKT would lead to inhibition of mTOR, reduction of protein synthesis, and increased protein breakdown that are very relevant in the context of sarcopenia. So what are toll-like receptors? They're a family of receptors that play a critical role in the immune system by activating inflammatory signaling pathways. The one we've been working most is TLR4, which is a receptor for lipopolysaccharide or endotoxin. Endotoxin is found in the cell wall of bacteria and activates TLR4 that is mainly expressed in immune cells, but also in other cells. And this triggers an inflammatory response and a whole host of immune responses. This is the structure of LPS, lipopolysaccharide, that has a core, also a glucosamine pyrophosphate polymer, and a lipid A. And this is very important because the long-chain fatty acids, particularly fatty acids and lipid A, are critical for its bioactivity. The TLR4 has two domains, an intracellular and extracellular domain. When it's activated, forms a dimer. It also attracts co-receptors, MD2 and CD14, and different adaptor proteins. And then once it's activated, it activates NF-kappa B, but also several MAP kinases as shown here. We became interested in this in TLR4 when Jeff Flyer published a few years ago data shown here that essentially linked TLR4 with the development of lipid-induced insulin resistance. And what he showed here using an insulin clamp, and these are data here from the clamp in which wild-type mice that are infused with a lipid develop insulin resistance, and shown here. Can you all see the pointer?
Yes, we can.
Okay, thank you. So lipid infusion induces insulin resistance, but in TLR4 knockout mice, there is no, or these mice are protected from the negative effect of lipids. Here linking TLR4 with lipid-induced insulin resistance. So we got very excited about that finding. And the first thing we wanted to do in our population here is whether there are abnormalities in TLR4 signaling in insulin-resistant subjects. So we took tissues from obese and type 2 diabetic subjects, and we measured TLR4 protein content shown here, and also TLR4 gene expression. And as you can see, there is a significant increase in muscle expression of TLR4, and also a direct correlation with the HOMA insulin resistance index. And this was the basis for us in focusing heavily on studying TLR4 and insulin resistance in the context of obesity and aging. So obese subjects have increased TLR4, but is this pathway activated by fatty acids? So from our participants, we grow new human myotubes. We stimulated with palmitic acid, different doses, and as you can see, a Western blot, palmitic acid induces a strong activation of this pathway. And using here a anti-TLR4 blocking antibody, we showed and others showed around this time that the effect can be blocked by the TLR4 antibody, is suggesting that the effect of fatty acids is at least in part mediated by TLR4. And we went back to our research participants and see, what are the major differences between this populations, lean, obese, and type 2 diabetics? So obviously, the diabetics have high glucose, have higher insulin, higher fatty acids, TNF, and IL-6. And we wanted to see what would explain the increased TLR4 expression. And the only one that from these factors that consistently increase TLR4 levels in muscle was fatty acids. And that is shown here again in primary human myotubes, showing that incubating myotubes with palmitic acid increases TLR4 levels in protein, also gene expression, and the effects can be reproduced by incubating them with lipid A, which is the part of LPS. To evaluate whether this could happen in vivo, we took lean healthy volunteers and we gave them a 48-hour intralipid infusion. And so they were lean. We expect them to have lower levels of TLR4 in muscle. So we brought them into the hospital for an intralipid infusion, and then we did an experiment control with saline and measure TLR4 in muscle. And as you can see, TLR4 was elevated by intralipid infusion, as well as other TLR, and in general, several gene, the expression of several inflammatory mediators was increased by the lipid infusion. I should highlight that we gave a very low dose of intralipid, 30 cc's an hour, and just caused a very small increase in fatty acids. We didn't want to give a very high dose that was supraphysiologic. So a small increase in fatty acid levels, which you would see, for example, in obese subjects, is sufficient to increase TLR4 levels. And we also did the opposite data I'm not going to show today. We took participants, obese participants with high TLR4 levels, high fatty acids. We gave them acipimox, which is an anti-lipolytic drug, and it reverted this effect. So we concluded that fatty acids acutely stimulate TLR4, but also chronically, they increase expression of TLR4 and then just causing chronic stimulation of NF-kappa B and MAP kinases. But around this time, as we were working on this hypothesis related to saturated fatty acids, this notion of this concept of metabolic inductoxemia was starting to emerge. And this is data. This is a nice figure from Dr. Neves that illustrates that. So essentially, as many of you know, the intestinal epithelium is a barrier that prevents the entry of endotoxins that are found, of course, in our microbiota in the gut. They prevent it to entering. But in certain diseases like obesity, high-fat diet, et cetera, endotoxin levels can enter the circulation and then reach several tissues where they can cause insulin resistance, inflammation in the blood vessels, and dysfunction of adipose tissue. There was also some evidence that this could be potentially prevented with prebiotics, et cetera. These are data from Dr. Dandona, where he had healthy participants, and his team gave them they challenged these participants with 2 different types of diets on different days, and a healthy American Heart Association type meal or a high-fat, high carbohydrate meal, and then measure the effect of a single meal on endotoxin levels. And as you can see, the high-fat, high-carb diet can increase endotoxin levels acutely. So we went back to our research participants samples we had stored from previous studies and measured endotoxin levels. And indeed, we were very excited to see that both obese and type 2 diabetic subjects have increased endotoxin levels. And there was an indirect correlation with insulin sensitivity measured by the M value. So these participants have high endotoxin levels, but does it mean that this endotoxin is bioactive? So we were interested in measuring its effects on insulin action. These are data on human myotubes that were first pre-incubated with endotoxin, then stimulated with insulin. And as you can see, over time, LPS causes a dose-dependent decrease in insulin signaling, stimulation, here measured with AKT and GSK phosphorylation. We use a glucose transport assay with a rat epitoclaris muscle to look at glucose uptake. So in this assay, we take the muscles from rats, incubate them, and then stimulate them with insulin and measure glucose uptake into the muscle. What we did is first we pre-incubated them with endotoxin, then stimulated with insulin. And as shown here, of course, insulin stimulates glucose uptake into the muscle, but a higher dose of endotoxin is able to reduce glucose transport, that's indicating that endotoxin not only is elevated in insulin resistant subjects, but also is bioactive and induce insulin resistance. So this model evolved that in obese subjects, type 2 diabetic subjects, and data and aging, which I'm not going to show today, they had two bioactive substances that can stimulate this pathway. One is fatty acids from a nutritional source, particularly saturated fatty acids, and the other one is endotoxin coming primarily from intestinal bacteria. And as we were working on this model, we wanted to test whether blocking signaling through TLR4 can prevent the inflammatory response and also the abnormalities in glucose metabolism. So we tested this compound developed by Takeda, TAC242, that prevents the docking of these proteins with TLR4 and is a potent inhibitor of TLR4 signaling, and evaluated whether it was developed for to inhibit TLR4 and inflammatory cells, but it wasn't known whether it would also work in muscle cells. So we used myotubes, and then we stimulated them with LPS, and TAC242 was also pretty effective in preventing the inflammatory response to LPS in muscle cells. How about fatty acids? So we stimulated the cells with stearic acid, which is, as you all know, another very abundant saturated fatty acids. And as shown here, stearic acid activates both NF-kappa B and JNK, and the effect is partially prevented by TAC242, although not completely prevented, which is opposite to the effect of endotoxin. How about effect on glucose uptake? So these are, again, the muscle cells stimulated with insulin and measured glucose transport. Here is maximal stimulation with insulin. So LPS reduces glucose uptake in muscle cells, and this effect is completely prevented with TAC242. Saturated fatty acids also reduce glucose uptake, and pre-incubation with TAC242 prevents the insulin resistance caused by stearic acid, although not to 100%. The effect on stearic acid is partial. So our model here is that the TLR4 completely mediates the effect of LPS on inflammatory responses and insulin resistance, at least in muscle. And TLR4 partially mediates the effect of fatty acids, but fatty acids must be working through other mechanisms. And as many of you know, there are a number of other lipid metabolites that have been postulated to induce insulin resistance in obesity, diabetes, and aging. So I've talked about obesity, insulin resistance, how about muscle? So in aging, and these are data from non-obese subjects. So we measured endotoxin in older healthy adults and reported that healthy aging is also associated with increased endotoxin levels. We also hypothesized that exercise could prevent the effect or reverse the effect of aging and endotoxemia, but actually turned out to have no really effect on endotoxin levels. So can endotoxin affect protein metabolism? And it does. And actually, this is something that pulmonologists have known for a long time because they're interested in the effect of endotoxemia on diaphragm function. These are some data we haven't published, but just showing that these are human myotubes, and the toxin also causes significant atrophy. And just interesting data from Dr. Rera and UCLA linking intestinal barrier dysfunction with aging, and they developed this assay called the Smurf Assay, in which they give a blue dye to the flies so they can eat it. And if they have intestinal barrier dysfunction, they turn blue. So that's why they call them Smurfs. So with aging, there's an increased proportion of Smurfs, demonstrating that there's a leaky gut with aging. But what's more interesting is that having intestinal barrier dysfunction early in life, as shown here in this line, can predict how long the flies will live. So in a way, intestinal barrier dysfunction predicts death. And it probably has to do with being a surrogate marker for stem cell function in the intestines. Okay, so we've been interested in moving this research into human translation, and been conducting clinical trials on this area for about 6-7 years. And we are just beginning to get sufficient information to begin reporting it. The idea here is to block either TLR4 signaling with an inhibitor, and we've done a clinical trial, and hopefully I can show those data in a different time. But today I'm going to talk more about our efforts in clinical trials for trying to reduce endotoxin levels in obesity and hopefully in aging in the future. So the first trial I'm going to talk about has to do with using two different strategies to try to reduce endotoxin. One is using Cevallon. Cevallon is a resin that is clinically used in patients with chronic kidney disease who are in dialysis, and they have high phosphate levels, which is a big problem in patients with chronic kidney disease. And Cevallon binds phosphate in the gut and prevents it from, and reduces levels systemically in these people. But it turns out that there are reports that Cevallon also can bind endotoxin in the gut and prevent or reduce its systemic levels. The other intervention was Bifidobacterium longum, as I'll show later. There are some data suggesting that it can reduce or improve metabolism. So these are data on Cevallon. I'm trying to provide some rationale why we used it. This is an assay. It's a measurement of free endotoxin level in the test tube. So simply demonstrating that Cevallon in the test tube can bind to endotoxin and reduce the levels of free endotoxin. There's also a large number of data in the kidney failure literature suggesting that Cevallon reduces endotoxin levels in this population, which have systemic endotoxemia. Regarding Bifidobacterium longum, we use a symbiotic, and symbiotics are treatments in which it combines a prebiotic and also with a substance like a probiotic that would stimulate the growth of particular bacteria. So we gave, in this trial, to our participants, Bifidobacterium longum as well as oligofructose. So the bifidobacteria and other bacteria can take the oligofructose and use it as a nutrient and be enriched in the gut. So these are data in rodents from Dr. Kani showing here that a high-fat diet causes glucose intolerance. This is a glucose area under the curve. Assay that cellulose has no effect, but in contrast, giving them oligofructose with bifidobacteria improves glycemia in these rodents. And there are some other data that provided rationale for testing these interventions in an attempt to improve intestinal function and also reduce endotoxin levels. So in this was a relatively small trial comparing the effect of placebo against sevelamer and against also a symbiotic, and we did it in two different populations, lean and obese subjects. We didn't want to add the confounding effect of aging here. Not that it's not important, but that would be the second step. So we first studied middle-aged participants and shown here, and of course, by definition, the lean subjects have a normal BMI. This is the study protocol. Subjects were received a low glucose tolerance test, different tests of intestinal permeability, stool collection for microbiome, and measurements of LPS and other markers of intestinal function. The insulin clamp is our main outcome for insulin sensitivity, which is considered the gold standard. And then subjects received for 4 weeks either a placebo, again, the symbiotic of oligofructose plus Bifidobacterium longum, or sevelamer. And then after the interventions, we repeated the outcome measures. So these are based on glucose and insulin sensitivity parameters. As suspected, obese subjects were non-diabetic, but they still have slightly higher plasma glucose, tended to have higher A1C. They were more insulin resistant based on the HOMA-IR index as well as the M value in the insulin clamp. They also had higher triglycerides, better HDL, and higher LDL levels, as one would expect. So did they have alterations in intestinal barrier dysfunction? And in contrast to what had seen before, the alterations were more subtle. They did have increased levels of LPS, but other measures of inflammation and intestinal barrier dysfunction were not seen in this population. It could be that in this stage of obesity and early insulin resistance, the abnormalities are milder, but indeed they do have higher levels of endotoxin. So what were the effects on the clinical outcomes? Starting with weight, interestingly, with sevelamer and symbiotic, there was small increases in weight in a month, just 1 kilogram, and we're puzzled about why that happened. Notably, it was not different compared with placebo, but nevertheless, it's an interesting finding. But sevelamer, but not symbiotic, did improve plasma glucose, and most importantly, it had a significant effect on insulin sensitivity measured by the clamp, improved by about 10 to 15%, which is about one would expect with other interventions such as metformin. So the effect is there and it's significant. However, the symbiotic had no effect. We took muscle biopsies in some of our participants to measure effects on insulin signaling. So as one would accept, this is measurements of AKT phosphorylation during insulin stimulation. Obese subjects have significant insulin resistance at the level of AKT, and there is a trend for an improvement on with sevelamer, although it did not reach statistical significance. Effects on cholesterol, along with improvements in glucose metabolism, there were also a number of improvement in cholesterol caused by Cevallon, improved LDL, increased HDL, and the LDL, HDL cholesterol. And I should know this is not entirely new finding. Others had previously reported that Cevallon can improve cholesterol. How is Cevallon working? Is it improving endotoxemia? And it turns out that it is not. The data on the top panel says in lean subjects, no changes in LPS or inflammatory markers or LMR. LMR is the lactulose-mannitol ratio, and this is a test in which one administers both lactulose and mannitol to research participants. And in general, when there are abnormalities in intestinal barrier function, the ratio will increase because lactulose would leak, but not mannitol. Surprisingly, even though Cevallon is improving a number of metabolic parameters, is not doing it by changes on LPS. So in top panels is lean subjects, and the lower panels obese subjects, and there is no change in any of these parameters. So how is Cevallon working? That's one question we wanted to know. And also, is the symbiotic administration sufficient to change bifidobacteria levels in the gut? So we did a microbiome assessment in the stool, and just very rapidly we used shotgun metagenomic approach in which we measure the levels of different of the taxonomic units, the individual bacterial species levels, and also gene pathways, the changes in gene pathways based on the metagenomes. Today, we're still mining this data. So today I'm just going to talk briefly about the changes in a specific bacteria. So, and these are data that many of you know. This is type of data many of you know. With taxonomic analysis, you get data on kingdom, the different phylum, and then several layers later, you get into individual species. So comparing lean versus obese, and the phyla level, as many of you know, the two main phyla that are more abundant in human gut are Firmicutes and Bacteroidetes. And the Bacteroidetes Firmicutes ratio is generally considered a index of health and the gut microbiome. And there are some data, although the data are somewhat inconsistent, that in lean subjects, the ratio is elevated as an indicator of health. But in this case, we did not see any difference. Granted that our sample size is not very large. There's obviously other much larger studies. So focusing on Bifidobacterium longum, here using a t-test, there was a tendency for a decrease in obese subjects, which is consistent with the literature, and also using a paired t-test. In the lean subjects, there is a tendency for an enrichment, but indeed in the obese subjects, administering Bifidobacterium longum increases their levels in the gut. So it's nice confirmation that at least our intervention is working to enrich Bifidobacterium longum. Measurements of alpha diversity and evenness, and in general has been reported that with obesity, there's a decrease in alpha diversity, and that's another measure of poor health in the gut microbiome. And consistent with the literature, we do see decreases in the obese subjects in terms of alpha diversity and evenness. And none of our administration of the interventions change these parameters. Now, focusing more in associations between the phenotype based on BMI and different species. So in lean subjects, there was a strong association with a number of individual bacterial species, and very importantly, Bifidobacterium longum, which is what we had hypothesized. But in addition, there are others that were associated with a lean phenotype, as well as others associated with a obese phenotype. These are data at baseline. So do the interventions change these? And Cevallon generally did not have an effect on bacterial species, nothing in obese participants, and then one species was changed in the lean subjects. Now, how about placebo? Placebo had changed the levels of only one species, Roseburia intestinalis, shown here, and notably, as I had mentioned before, their Bifidobacteria longum did change in response to the symbiotic, and also to other species, Roseburia and Holdemania. So again, it's good to see that our intervention is having the expected effect on the microbiome. But again, how is Cevallon working to improve glucose metabolism? Is not working through endotoxemia, since levels didn't change, even though they are modestly elevated in obesity. They were not improved by Cevallon. Doesn't seem to have a significant impact on the gut microbiome. So it must be doing something else. So we used an unbiased metabolomic approach. So we sent samples to Metabolism, and they measure samples, the level of several thousand metabolites by mass spectrometry. And we only sent the samples for Cevallon, since the symbiotic did not have a significant effect on metabolic outcomes. We did not do those measurements. So we were restricted to the effect of Cevallon and the placebo. So first, comparing lean and obesity, and again, these are, again, is the effect of on plasma metabolites. 85 metabolites increase higher in obese subjects and 15 in lean subjects. And we also evaluated the effect of the intervention placebo and Cevallon. The placebo, notably 88 metabolites were decreased, and which is a very interesting finding, significantly decreased, and most of them were different types of fatty acids. And Cevallon had effect on several metabolites, primarily increasing 109. I'm not going to go through this in detail, just in the interest of time. Just to know that in obesity, many of the metabolites that were elevated have also been reported before. Many amino acids, number of lipid metabolites, fatty acids, acylcarnitines, DAX, ceramides, phospholipids, etc. And there were some metabolites increase in the lean, and I will talk about that in the next few slides. So mining these data, that it of course takes time, but it's actually fun, because one goes back and trying to relearn biochemistry and metabolism. So these are different metabolites that were increased in obese subjects. They were reduced by Cevallon and not reduced by placebo. And I'm going to go over them. One of them is 4-hydroxyglutamate, and this is a glutamate metabolite. Of course, glutamate is a very important neurotransmitter and has a number of other important effects. And I'm becoming interested in this because there are very large metabolomic studies, and some of them have linked high levels in preeclampsia, which of course this is an important problem in pregnant women. And also notably, it's decreased by metformin. These are not our data, but data from other data sets. So that's turning out to be an interesting lead. Another one is 5-hydroxyindole sulfate, which is a tryptophan metabolite. Again, tryptophan, a very important amino acid, a precursor for serotonin, niacin, melatonin. So that could be an interesting lead. Another one is 10-decenrate. I'm not sure if I'm pronouncing it right. Maybe some biochemist in the audience can correct me, but this is an unsaturated fatty acid. It's a plant metabolite. It turns out to have potent antifungal properties. I don't think that's how it's working in this study, but it's just an interesting piece of information. The other one is methyl palmitate, which is a fatty acid ester. It's a potent muscarinic antagonist, and it's also a potent vasodilator. The next one, the next set of data is metabolites that were reduced in obesity. They were increased by sevelamer and not increased by placebo. One of them is ascorbic acid 3-sulfate, which is a metabolite of ascorbic acid. And as you all know, it's a very potent antioxidant. Now, I'm not that interested in this one, at least in the context of insulin, glucose metabolism, because there's a lot of studies testing ascorbic acid in humans and insulin sensitivity, and the data tends to be inconsistent. Other metabolites that changed were a few amino acids shown here, but the one that I'm also becoming more interested is in hypotaurin. Hypotaurin is cysteine metabolite and a precursor of taurine, and it's a potent neurotransmitter, also a potent antioxidant. And there's data that it reduces fatty liver in rats, and that it also increases lifespan in C. elegans. And of course, it's important for bioenergetic metabolism. There are some data that hypotaurin can stimulate the growth of cancer cells. So not all the data on this metabolite would point towards having benefits, but I think still is a very interesting lead. Now, some of you may know that sevelamer is also a bile acid binding resin. There's some data suggesting that, and of course, bile acids have profound effects on glucose and lipid metabolism. So this is a cartoon or a figure I adopted from Hendrickson, just going back to some basic physiology and bile acids. So bile acids, when they are increased inside the gut, the enterocyte, they get activated through the FXR receptor, and then they stimulate the release of fibroblast growth factor 19 into the blood, FGF19. And FGF19 binds to its receptor in the liver, and its binding causes a reduction of bile acid secretion. So in this model, bile acids, it's a negative feedback mechanism by which bile acid synthesis is regulated. So one way to see the effect, the endogenous effect of a drug on bile acids is by measuring FGF levels in the blood. And if they're decreased, it suggests that FXR is being activated at the level of the enterocyte. And indeed, we see a reduction of FGF19 caused by sevelamer, again, indicative of bile acid binding in the gut. And I should highlight that these type of data have been reported a couple of years ago by Dr. Knopf in Germany. Looking into the effect of bile acids in the blood, he researched data on thyrocolate, one of the most abundant primary bile acids in our study, demonstrating that sevelamer is increasing thyrocolate. And other bile acids also increased, although this one was the most significant. So another potential mechanism by which Cevallon is improving insulin sensitivity is shown here. So there's sequestration of bile acids, reduction of FGF19. This leads to the repressive effect of FGF19 and its receptor is removed. So that leads to an increase in bile acid production and release into the circulation. Bile acids bind to its receptor in TGRF5, which is a bile acid receptor in muscle, and this could lead to an improvement in insulin sensitivity. So this is one of the models we're pursuing, as well as some of our other leads that I mentioned. So to summarize, obese subjects have metabolic endotoxemia. I think that the more severe the obesity and the diabetes is, more likely that or more severe the endotoxemia will be, and as well as aging that has an independent effect. Obese subjects indeed have differences in the abundance of B. longum in the gut, but neither Cevallon and Bifidobacterium longum administration improve circulating levels of LPS in obese subjects. So we're still in the search of a good intervention to reduce LPS levels in human subjects. So if anyone has a tip on something else we could do, please let me know, because we think still this hypothesis, metabolic endotoxemia hypothesis, has not been fully tested with our experiments. Obese subjects have differences in the abundance of several bacterial species in the gut, in addition to B. longum, but enrichment B. longum with a dyssymbiotic did not improve any metabolic parameter. So the good news is that while B. longum does not seem to be, at least with our data, a good target, there are many others, and our data and data from many other groups have shown differences. Here I just showing again the data on of differences between the lean and obese phenotypes. And my sense is that a single bacterial species approach won't work. My sense is that many will need to be targeted at the same time. Cevallon improves glucose concentrations, insulin sensitivity, and cholesterol, but the beneficial metabolic effects of Cevallon are not due to changes in endotoxin, intestinal permeability, or the microbiome. But Cevallon does have a multitude of previously unknown effects on levels of many metabolites, and we think these could be responsible for improving glucose and lipid metabolism, and identifying these, which is the mechanism or mechanisms by which Cevallon work could lead to novel ways to treat glucose and lipid metabolism abnormalities in obesity, diabetes, and aging. So with this, I'm going to finish. A lot of people from my lab and our clinical research centers shown here have contributed to this work. The Jocelyn, Alex, Kostic, Yang Zheng, and Log Juyan Pham have helped us with the microbiome analyses. Dr. Kyung-Feng Dong with the bioinformatics is at Loyola University. This work supported by NIH. The microbiome study supported by ADA, GSK, support from the VA, and the symbiotic and oligofructose were kindly provided by Lallemand, which is a company that generously gave us the symbiotic. And with this, I'm happy to take questions. Thank you.
Thank you very much, Dr. Musi, for an excellent presentation. For folks who have questions, please submit them through the Q&A function, and I will read them. So there's already a few coming in. The first one is from Dr. Mazepina. It asks, do people with leaky gut and IBS have greater risk for developing insulin resistance?
In general, yes, there is. an association between leaky gut, endotoxemia, and insulin resistance. Yes. I don't know specifically in the context of IBS. Of course, the number of diseases of the gut and the GI system are associated with leaky gut and endotoxemia, and one would think they're also associated with insulin resistance. I don't know in the particular, in IBS in particular, but it's a very good question that it is worth pursuing.
Okay. Dr. Lustgarten asks, intestinal alkaline phosphatase has shown promise as a means to prevent metabolic syndrome in mice. When considering that intestinal alkaline phosphatase dephosphorylates LPS, what do you think about using IAP as an approach to reduce or prevent endotoxemia?
Oh, I was not aware about that. I think it's a great idea. I think it should be pursued, definitely.
Okay. Dr. Zhandong Wang asks, as you mentioned, Cevallon is a non-absorbable bile acid sequestrant for the treatment of hyperphosphatemia in patients with chronic kidney disease. Can you study if patients on hemodialysis who are treated with Cevallon exhibit lower serum levels of LPS? So in kidney disease patients, do they have lower LPS with Cevallon?
Yes, that's a very good question. And actually, the research on Cevallon and endotoxin has been really initiated by nephrologists treating their patients when on dialysis. And the data does point, including small and larger studies, that Cevallon reduces endotoxin in patients in dialysis. That was actually one of the reasons, but rationale why we proposed our study. Even though we were not looking at participants with chronic kidney disease. I should also say that it's in HIV, these patients also, even healthy patients with HIV have high levels of endotoxin, and it's thought that this is one of the reasons why they have higher prevalence of metabolic disease and cardiovascular disease. And large trials have been done to target endotoxin with Cevallon. And in contrast to what has been shown with patients with hemodialysis, in patients with HIV, Cevallon did not reduce endotoxin, which is along with what we've seen. And of course, these data is relatively recent. So when we started 7 years ago, we didn't know about this, but yeah, so the data is, I say, inconsistent, and hemodialysis Cevallon seems to be very effective, but not in other diseases like HIV and maybe metabolic disease.
Thank you. Dr. Alice Lichtenstein asks and compliments you on a fascinating talk, but with regard to the free fatty acid increasing TLR4, could you be more specific about differences among specific free fatty acids? Is there a clear difference between saturated and unsaturated, for example? Have you studied trans fatty acids or?
Absolutely, yes. So there's a lot of data. In general, it's thought that saturated fatty acids are very potent to activate TLR4, but not unsaturated fatty acids. Not only that, but polyunsaturated fatty acids do not activate TLR4, but actually prevent the negative effect or the inflammatory effect of saturated fatty acids. I should also, for the sake of giving a balanced view, there are also some data that do not have not support this notion that the activation of TLR4 by fatty acids plays an important role in a major role in metabolic disease. I think since we started working on this, if I had to guess, I would say that 2/3 of the data point towards a TLR4 having a role through saturated fatty acids, and maybe a third not supporting this hypothesis. But then again, the same with many other theories in terms of metabolic disease.
Thank you. Dr. Andy Greenberg asks, and actually I was thinking about this, very good question, Andy. In terms of the literature, microbial production of small chain fatty acids by microbiota, that's been around for a while, right? But he just says not as much focus on endotoxin, but did you measure small chain fatty acid levels? I kind of thought that's an interesting question too.
Yes, that's a very good question. And so we did not, because, well, we did measure them not in the gut, we did it in plasma. And we measured them in the lean obese participants and the placebo and Cevallon. We didn't do it with the symbiotic, and again, was just a cost efficiency approach, because since we didn't see major effects on metabolism, we kind of stopped there. So we did not measure short chain fatty acids and effects from the symbiotic. We did with Cevallon, and there were no effects. Cevallon did not affect them.
Okay. Let me just maybe check the chat too. Sometimes the chat comes in with the questions, but it doesn't look like any in the chat. So Nick, what about the effects of weight loss on endotoxemia? I mean, maybe that's sort of obvious, but I just wonder if if you eliminate the obesity or reduce weight and body fat, if there are effects.
That's a very good question. I don't know, and maybe someone else may have the answer here in this audience. I think the data is pretty good, at least in rodents, and there's some data in humans too, that weight gain and fatty acid and fat administration, high fat feeding, causes an increase in endotoxemia. I don't know about the opposite, but it's something that could be done easily in people that have samples from weight reducing interventions. And I'm trying to think if there's literature on this, but I can't recall. But if someone has samples, that would be relatively easy to do. The only thing is, it's a little tricky to measure endotoxin in plasma, because there are a lot of inhibiting factors in plasma. So one has to neutralize them with heat inactivation, and there's also there's endotoxin everywhere. So the assay could easily be contaminated. So one that needs to be careful about how the samples were obtained and processed. Another way to do it is measuring LPS binding protein, which is it's a good surrogate marker of LPS levels. And of course, it's much more stable and can be measured by an ELISA. So that's another good way to do it. So if anybody has samples, I would encourage them to measure it, or we'd be happy to do that.
Great. So one other question on the Q&A from Dr. Mathan. In addition to plasma bile acids, did you have an opportunity to examine fecal bile acid excretion and fecal secondary bile acids production following Cevallon administration?
Yes, we haven't done, we have the stool samples. So we haven't measured bile acid levels in stool, which is something we could do. Yeah, secondary bile acids did not change significantly with Cevallon.
Okay. and
Maybe,
then
maybe one of them, I can't recall, but the effect was small.
Okay, great. And we have time for one final question. What was the rationale for the dosage of Cevallon? You used 1.6 grams 3 times per day. Any protective effects of Cevallon could be achieved without any adverse effects such as hypophosphatemia for long-term use.
Yes, the rationale, we just use a dose that is used in patients with chronic kidney disease. So that was easier to justify from a regulatory standpoint. It's generally very well tolerated. It can cause a little bit of constipation, a little bit of mild gastrointestinal adverse events. We didn't see them in our participants, even though it was small. There has been reported cases of severe intestinal obstruction, but those are very rare. And of course, this is a drug that is given to many thousands of people in the dialysis world. In people with normal phosphate levels, Cevallon will not reduce them. We knew that from before, and we did test phosphate levels, and they did not change.
Okay, well, just again, on behalf of the HNRCA, Nick, I just want to thank you for an excellent seminar, and thank you to all the attendees and all the great questions. This concludes this afternoon's seminar series. Thank you. Okay, so we're out of the